Hello everyone, my name is Yousef. I am an undergraduate student at the University of Utah. I will be presenting my work with my advisor Skyler Jennings on simulating the human compound action potential using a physiologically realistic model of the auditory periphery. As you might know, electrocochleography is a technique used to record the electrical potentials generated by the inner ear and the auditory nerve in response to sound stimulation. In our lab, we record these potentials non-invasively from an electrode placed on the tympanic membrane, with the stimulus being presented in alternating polarity. We subtract and add these different polarities to emphasize the hair cells versus the auditory nerve contribution. Here's an example of a recorded cochlear response elicited by a brief tone burst. Such a response can be decomposed into three main potentials. The first is the cochlear microphonic, the second is the summating potential, and the third is the compound action potential. The focus of today's presentation is the compound action potential, or CAP for short. I will first describe our approach to simulating the CAP, and then I will compare our simulations with human data collected in our lab and also from the related literature. So our approach to simulate the human CAP was based on the convolution model proposed by Goldstein and Kiang in 1958, and here I will briefly describe the model. Here is a simplified diagram of an uncoiled cochlea showing the innervation of the auditory nerve fibers at various characteristic frequencies between the base and the apex of the cochlea. When stimulated by a sound, each one of these fibers will fire an action potential, and the firing pattern that occurs at 1 CF is quantified in what is known as a peristimulus time histogram, or PSTH for short. This histogram shows the firing rate per unit of time at 1 CF. Now, what Goldstein and Kiang proposed was a convolution model that treats this peristimulus time histogram as an input, and the output of this model is the predicted CAP. This convolution model is characterized by what is known as a unitary response, which can be thought of as an impulse response of the system describing the changes that occur to the peristimulus time histogram between the auditory nerve and the electrode sites, which are commonly located at the round window or the tympanic membrane. I should emphasize here that in our simulations, we took into account multiple peristimulus time histograms to account for all the CFs in the cochlea in order to get an accurate prediction of the human CAP. As I mentioned, the convolution model amounts to convolving the unitary response with the peristimulus time histograms obtained at each CF. Our simulations incorporated the unitary response described by Ilperling in 1976, which was derived from data based on high-pass masking technique performed on human subjects. We simulated the peristimulus time histograms of 221 CFs ranging from 250 up to 20,000 Hz using the synapse output from the Zilani et al. 2014 model with the human cochlear tuning and normal outer and inner hair cell functions. Further, high, medium, and low spontaneous rate fibers were simulated for each CF, and the responses were weighted according to the distribution of spontaneous rate fibers described by Lieberman in 1978. Finally, to get the CAPs in units of microvolt, the peristimulus time histograms obtained from the model were scaled to approximate the potential observed at the tympanic membrane electrode according to data from Anatoly Candela and Kiang in 1978. Note that the unitary response is level independent, and thus level dependent changes in the CAP morphology or nonlinear CAP amplitude growth originate from nonlinearities already accounted for in the Zilani's model. Here's a comparison between the human CAPs.
elicited by 100 microsecond acoustic click collected in our lab, and the predicted CAPs obtained by the convolution model. We see that the model qualitatively follows the level-dependent morphologies of the human CAPs. Specifically, the model predicts A, the increase in the width of the N1 peak of the CAP as we decrease the intensity of the click, B, the appearance of the second positive peak, especially at high click intensities, and C, the increase in the amplitude of the CAP and the decrease in the latency of the CAP as we increase the click intensity. Here we show a quantitative comparison between the human and the model predicted CAPs in terms of the amplitude and the latency. We see that the model captures the growth in the CAP amplitude, including regions with rapid growth at low and high click levels, as well as shallow growth at moderate click levels. Similarly, we see that the model predicts the decrease in latency as we increase the click levels. However, the predicted latency change at a slower rate as a function of the intensity compared to the human data. So the overall change in latency in the human data is greater than the predicted ones. As you might know, a hallmark of synaptopathy is the reduction in the amplitude of the CAP, or wave 1 of the auditory brainstem response. Here we show that our model predicts the reduction in the CAP amplitude when we exclude the low and medium spontaneous rate fibers. The left panel here shows the predicted CAPs that are elicited by clicks and their amplitudes and latency. Here we included all types of fibers, low, medium, and high spontaneous rate fibers. The right panel here is the same simulation, but only including high spontaneous rate fibers. This dashed curve here is exactly this curve here. We see that as expected, suprathreshold CAP amplitudes are reduced, while amplitudes near the threshold remain unchanged. Here we compare click versus chirp evoked CAPs. A click stimulus is expected to result in an asynchronous stimulation of the auditory nerve fibers in the cochlear base and apex due to the delay imposed by the traveling wave. Rising frequency chirps have been developed to account for this delay and to produce greater synchrony across fibers of differing CFs. On the left, we have the predicted CAPs that are elicited by clicks at different intensities and their amplitudes and latencies. On the right, we have the predicted CAPs that are elicited by chirps with varying intensities. The black thick curves here are the amplitudes and the latencies of the predicted CAPs that are elicited by chirps. The black dashed curves here are the amplitude and the latencies of the predicted CAPs that are elicited by clicks. So they're exactly these curves here. The red curves here on the top and the bottom figures are the measured amplitude and latencies from human subjects reported in Chertoff et al. 2010 for the CAPs that are elicited by chirps and clicks. We see that the model is qualitatively similar to the human data and that we see a larger amplitude in the case of the chirp. However, the model predicts a larger chirp benefit than observed in the human data. Additionally, the model incorrectly predicts an increase followed by a decrease in the amplitude as we increase the stimulus level, which is not observed in the human data. As to the latency, we see that the model correctly predicts a slower rate of decrease in the case of the chirp stimulus. Here we will look more closely into the contribution of individual CFs to the predicted CAP. On the left here we have the predicted CAP evoked by a click, and on the right we have the predicted CAP evoked by chirp. These plots here are the individual peristimulus time histograms obtained at each of these CFs. The black, red, and cyan histogram correspond to high, medium, and low spontaneous rate fibers, respectively. The plots in the middle are the population peristimulus time histograms, which are obtained simply by summing up individual peristimulus time histograms across 221 CFs. And on the top panel, we have the predicted CAP. Consistent with our expectation, the chirp compared to the click results in a greater synchrony of neural firing, which leads to a larger and sharper amplitude in the predicted CAP. When we increase the chirp level to 110, we see that the low frequency segment of the chirp stimulus result in this upward spread of excitation that adapt high CF fibers, and this upward spread of excitation leads to the appearance of this early latency component in the predicted CAP. This adaptation weakens the response of these CFs when the instantaneous frequency of the chirp reaches the CF of these fibers. We were unable to determine the extent to which this early latency component is present in the chirp evoked CAPs in human, based on the current available literature. So an amplitude-modulated stimuli may evoke phase lock-in of the auditory nerve fibers in response to the stimulus envelope. An example of an amplitude-modulated sinusoid is at the bottom of the slide here. These waveforms are the average CAPs elicited by an AM carriers from 8 normal hearing subjects. These data are qualitatively similar to the envelope following response, EFR, that are recorded from an active electrode placed on the higher forehead. However, unlike an EFR response, which peaks at around 40 Hz and drops in amplitude after 80 Hz, these CAPs that are recorded from the tympanic membrane exhibit a robust amplitude across a wider range of modulation frequencies. See that the model predicts the largest CAP that occurs at the onset of the stimulus. It also predicts the adaptation that occurs after that. Moreover, the model predicts the increase in the first CAP amplitude as we increase the modulation frequency, which is consistent with the human data. To further assess the model predictions with regard to the AM stimulus, we compare the spectra of the measured and the predicted CAPs. We know that the human data is characterized by a spectral component that occur at the modulation frequency, as well as harmonics of that component. We see that the model predict these components as well as additional components that are not observed in the human data due to noise. This tells us that the model highly captures the morphology of the CAPs observed in the time domain. We have a plot of the spectral magnitude of the model and the human CAPs at each modulation frequency. Like the human CAP, the model predicts a robust phase lock-in across a wide range of modulation frequencies, where the largest magnitude occurs at around 160 Hz, and then slightly declines at higher frequencies. Here we show the contribution of individual CFs to the predicted CAP evoked by 80 Hz amplitude modulated carrier. We see that phase locking to the stimulus envelope is generally weak at the 3000 Hz carrier frequency and is relatively stronger for CFs higher and lower than this frequency. This implies that the CAPs evoked by a tonal AM carrier are predicted to emerge from off-frequency stimulations. In summary, our simulations support the following. The human CAP emerges from synchronous auditory nerve activity. It depends primarily on neural activity from the cochlear base when evoked by a click. It includes basal and apical contributions when evoked by a chirp. It has nonlinear morphology that originates from cochlear and auditory nerve nonlinearity, not from level-dependent changes to the unitary response. It is primarily the result of high spontaneous rate fibers.
It has reduced suprathreshold amplitudes when low and medium spontaneous rate fibers are absent, consistent with synaptopathy. And finally, it exhibits robust phase locking to amplitude modulation across a wide range of modulation frequencies. Some of our future directions include predicting derived band CAP from high-pass Maskin experiments, simulating the effect of eliciting the medial olivocochlear reflex on CAPs measured in quiet and background noise, designing a novel chirp stimulus based on optimal model predicted synchrony. And thank you.